88 of the Stem Cell Podcast, Cerebellar Organoids, with Dr. Giorgia Quadrato. Hello, everybody. We are Doctors Daylon James and Arun Sharma. Welcome back to the Stem Cell Podcast, where we culture knowledge in stem cell research by talking to some of the brightest minds in the field. The podcast is brought to you by STEMCELL Technologies, a global biotechnology company supporting life science research and fostering communication and collaboration in science. If you're enjoying this listen, please rate us and leave a review. We're always looking for feedback on how the podcast can be improved and for suggestions on guests. Today, as I said, we have Dr. Giorgia Quadrato. She's on the podcast to talk about her research on brain development and disease. We've also got our usual roundup of recent highlights and stem cell news that's coming right up.
But first, ISSCR 2025 is coming up. The meeting is taking place June 11th to the 14th in Hong Kong, and you can submit your late-breaking abstract until March 19th. Check out isscr2025.org to learn more. Definitely can't wait for ISSCR just in a few short months. We'll dive right into the roundup first here, though. We've got a nice symmetry going on in this roundup. Today, we've got two hepatocyte-centric papers and then two cortical organoid brain/papers to lead us into our guest today, Giorgia Quadrato. We'll start things off with a Nature Genetics paper. This is coming from a listener of the show, a friend of the show, Moritz Mall. His lab is out there at the Cell Fate Engineering and Disease Modeling Group at the German Cancer Research Center in Germany. And we actually ran into him last year at ISSCR. I don't know if you remember that, Daylon. We caught up with him with a few minutes. He actually used to work in Marius Vernig's lab. Of course, we just had Marius on the show a couple episodes ago. Check that out. And so the title of this paper is Active Repression of Cell Fate Plasticity by PROX1 Safeguards Hepatocyte Identity and Prevents Liver Tumorigenesis. And Moritz, going with the theme of the Vernig lab, and like I said, that's where his training came from, the Vernig lab and the Mall lab now. It's really interested in this idea of cell plasticity, cell fate, inner conversion. I mean, Marius had that amazing paper a while back that we talked about on the show, converting fibroblasts into human neurons, direct reprogramming, direct conversion. And this is a paper that's in that same realm. So cell fate plasticity is something we've talked about a lot. It's thought to be more prevalent or highly prevalent during development and actually enables development in a lot of ways. But plasticity is also a cancer hallmark, unfortunately. There's cell conversion, interconversion from non-cancerous states to cancerous states, and so on, right? And while master transcriptional regulators actually are able to induce lineage-specific genes to restrict this plasticity, to actually limit the interconversion, it's not totally clear whether this plasticity can be suppressed actively by lineage-specific repressors. And the lineage that they're focusing on here is the hepatocyte lineage. And so what the Mall lab, and the first author here is Bryce Lim, what these folks did is computationally predicted what they called so-called safeguard repressors for 18 different cell types that blocks phenotypic plasticity lifelong. So you can these factors are supposedly able to block that interconversion throughout the entire lifespan of the organism. And that's the computational side of it. And then they dove in more on the hepatocyte side of things and validated hepatocyte-specific candidates using reprogramming, actually showing that a specific homeobox protein, Prospero homeobox protein 1 or PROX1, enhanced hepatocyte identity by direct repression of alternative fate master regulators. And then they took it to from the in silico, from the in vitro to the in vivo. In mice, they showed that PROX1 was required for efficient hepatocyte regeneration after injury and was also sufficient to prevent tumorigenesis in the liver. So there's the disease model right there. And also in line with some patient data, PROX1 depletion caused the hepatocyte fate loss in vivo and enabled the transition of hepatocellular carcinoma to a different type of carcinoma, cholangiocarcinoma. So this is a really nuts to bolts paper going all the way from in silico, in vitro, in vivo, and even some patient validation. And then the last element of this was overexpression of this PROX1 actually promoted the cholangiocarcinoma back to the hepatocellular carcinoma transdifferentiation. So it's like kind of a yin yang back and forth sort of approach. So ultimately, they narrowed down PROX1 as a hepatocyte specific safeguard in terms of preventing, regulating this interconversion, this plasticity. And they support a model where cell type specific repressors can suppress plasticity throughout the entire lifespan of the organism to actually safeguard lineage identity and prevent disease. So there's a lot going on in this Nature Genetics paper. I'm actually curious about why they put this in Nature Genetics because I think there's so many aspects to the story in terms of development, in terms of disease, but a really high profile paper here going, like I said, from in silico to in vitro to in vivo to some clinical validation as well, ultimately to show this master regulator of lineage specification and plasticity in hepatocytes in PROX1. A really, really great story from the Mall lab.
Yeah, this is a great story. Nature Genetics is big up. That's why I went there, Arun. But I do agree with you. I don't look at Nature Genetics generally because I don't find a lot of stem cell or like cellular plasticity transdifferentiation stories, although maybe there should be more, right? The genetic bases of cancer and transdifferentiation are probably myriad. Here, the story is fascinating to me on many levels. One, all you vascular heads out there will recognize PROX1 as a sinusoidal endothelial marker. So I'm kind of like, oh, what's necessary in the liver? Right, yeah, because sinusoids in the liver play a huge role. But is it even doing that? So like, I don't know what the connection is there, but it's always interesting to me and a surprise to see how nature uh, reuses things for many different purposes, and this may be a case of that. Also, though, I love this from like I'm getting to the stage here, I guess, which is to say I'm circling the drain, maybe. That I recognize that the the people behind the stories and how they relate to their mentor and the mentor's mentor. And we just had Marius on the show, and we talked about a little bit about this. And here you can see how, that kind of, the neuro tree thing where you see all the scientific ancestry, all the mentorship and all that. This is such a great example of how the feed forward mechanism where you can be influenced heavily even to the point. This is not at all derivative, but the point I'm making here is that this is within that kind of wheelhouse of transdifferentiation and self-fate control. But here, you know, twisted toward a different end. I shouldn't say twisted; it makes it sound bad, but you know, aligned toward a different outcome. And I think really an exciting illustration of how the biology of cell plasticity, which really was more of like a kind of a pragmatic approach to getting to the cell types we wanted in the stem cell field, is really unveiling a lot of these mechanisms of disease pathology. So I love this from a kind of PI angle of how you see a PI coming out of the shadow of his mentor and creating his own imprint, their own imprint, in a really fantastic, exciting, and fascinating way. And that's my take. Many takes.
Yeah, absolutely. I totally agree with you. Shout out to Moritz here for such a really cool story. I like what you're getting at about the evolution of a trainee and how the differentiation, I suppose, from that trainee from the ancestral lineage of the mentor. It's a nice parallel that we've got going here. I like, I hope you like what I did there.
Yeah, that
Um,
was good.
yeah, yeah. So that was cool. And in terms of the nature genetics thing, I agree with you. I love nature genetics. I'm not hating on nature genetics at all, but you're totally right. As stem cell biologists, as cell biologists, it's not always our first journal that we look at when it comes to stem cell biology specific papers, but really high quality work here. I was just curious about why they didn't go to like Nature Medicine or Nature Cell Bio, which I'm sure would have loved to take a paper like this, but regardless, an amazing study, and shout out to Moritz again for this awesome work.
Well, Nature Genetics trying to get into the stem cell game. Here's the first deposit. Maybe you guys leave, what we did for DevCell. We'll take a big fat brown envelope. I'll leave my address on the website, and maybe we get some more Nature Genetics papers on the program. Just kidding. We're not for sale. Moving on to the next story here, we got another, as Arun alluded to. We're doing a beautiful symmetry here in today's episode. And I'm gonna mirror Arun here with my own hepatocyte story. This is a more like, again, I'm kind of wandering out of the stem cell field, but talk about stem cells, you got to talk about the liver. It's a major hub of stem cell activity and regeneration, just like in the normal physiological steady state, right? But here we're talking about a kind of a gene targeting approach, which I think the intersection of genetics, genomics, engineering, and stem cells. It's obvious. And there's a lot more stories, I think, that are combining those two elements. In this case, we're talking about liver and liver disease, genetic disease or other. But the hook here is that the liver is like the sink for the body in terms of gene targeting, right? Really amenable to gene delivery. In fact, if you have, like, let's say, some kind of overexpression vector that you want to be expressed systemically, then you put it in circulation, it ends up in the liver, right? Famously, lipid nanoparticles, it all piles up in the liver like 100-fold more than other organs for obvious reasons. This is the filtration mechanism for our health. But it's, can be a problem in that in terms of targeting disease, there's a lack of specificity, right? And when specifically talking about like disease targeting. So the liver's really amenable to gene delivery. And a lot of trials have showed really impressive results, namely with the CRISPR-Cas9 and base editors that have been aimed at like disruption of genes, like, for example, transthyretin, PKSK9, which I'm not going to tell you the name of that, look it up. But these are all mechanisms that are focused on disruption, right? You can break the system. We talk about on the show a lot. It's easy to break something, but to fix it, it's tougher. A lot of liver diseases are based on repair as opposed to disruption. And again, you can use the base editors toward that end, but it can be really tough because the mutations that underlie even a common disease, the specific mutation, point mutation or other, can be, unique to each patient, and you can't really customize your gene vectors. It's not really practical, although possible. So this was an approach that was based on targeting cells and then giving them a selective edge. And this leverages the capacity of the liver that has tremendous regenerative potential, right? So you count on the liver to kind of restore itself and copy all the cells that you've targeted to express these therapeutic transgenes. And here, in a story from Gang Bao and William Lagore, who are from Rice University and Baylor College of Medicine, respectively, they used this approach where they coupled this genetically encoded survival advantage to the expression of this therapeutic transgene. Specifically, they call this thing repair drive. And what it does is it transiently conditions the liver by knocking down this essential gene, which is fumarole acetoacetate hydrolase, otherwise known as FAH. And then it also delivers an untargetable version, right? So it knocks down the native and it delivers this version that can't be knocked down. And that not untargetable one is delivered coupled with the therapeutic transgene. And what they show is that when they use this approach, they can increase the percentage of targeted hepatocytes in a wild type mouse up to 25%, which is a ton. I mean, it's a lot. When you're talking about therapeutic load, you can get up to like 5% or less for some conditions of restored liver function in order to, get over the hump with the disease. So 25% is massive. And it resulted in a five-fold increased expression of this transgene human factor 9, which I presume is being used for hemophilia. And there was no toxicity, no tumorigenesis. And I think the key here, why this was a science translational medicine story, is that they went out to 1 year in the follow-up. This is all in mice, but I think that this is a preclinical model that is going to move to monkeys and could be a really valuable tool, not just for this specific condition, but any kind of disease in the liver or maybe outside of the liver even, maybe not to the degree that the liver can recover and regenerate, but there may be other tissues where this approach, this kind of, giving a selective advantage vis-a-vis the whole chimerism approach that's been done, in order to incorporate more a larger percentage of target cells into the animal. So an exciting technical approach, not necessarily a stem cell story, but stem cell adjacent. Arun, what do you think?
Yeah, we're doing a lot of stem cell adjacent stories on the show these days, all different types of tools. So this is right in our wheelhouse. This is a nice approach. Liver is thought to be one of those target organs for a lot of diseases. You mentioned PCSK9 inhibitors and all these kind of things. PCSK9 has taken the cholesterol field by storm. There's been tropism for AAV that has gone to the liver, AAV8, for example, with pros and cons. I mean, so that's almost like an unintentional consequence of that delivery mechanism, the AAV. But here we're talking about more targeted delivery. And I like what you got to at the end there. What excites me is in addition to the cool name of this thing, repair drive, it's almost like a sci-fi name, right? A hyperdrive, repair drive. Anyways, but yeah, in addition to the targeting of the liver here, it'd be really cool to extend this to other highly proliferative tissue types. I don't know, pancreas came to mind because there's really a need for targeted delivery for therapeutics there. But that's the thing that really excites me here with this story. Really cool work.
Yeah, and I love a hack. I mean, this is, I think, all the best, not the best, I don't want to qualify them, but all the most practical and effective, pragmatic approaches, I think, often involve a hack. And this is one of those. And I love it, the creativity involved and how you can leverage, as you're alluding to, sometimes it can be a deficiency. And I think a lot of times people have talked about this, like, oh, the liver. The problem is with gene delivery targeting, it all ends up in the liver, lipid nanoparticle, all ends up in the liver. But like here, let's use that. And I think that this speaks to the creativity and pragmatism of scientists that I really love. And I'm so proud of my peers.
Likewise, likewise. All right, shifting gears to the second half of the roundup here. Like we said, we've got some really nice symmetry. The first two stories are focused on liver, hepatocytes. The last two stories are going to focus on the brain and the cortical side of things, organoids, cortical organoids, leading into our guest here today. So the paper I'm going to talk about is actually a pretty short report. It's a clinical and translational report from Cell Stem Cell. And I know Sheila Chari, the editor of Cell Stem Cell, loves these clinical and translational reports these days. There's quite a number of them in Cell Stem Cell now. This is a preclinical safety and efficacy of human iPS derived products for, importantly, autologous cell therapy in Parkinson's disease. Now, this is a topic that we know a lot about. We've talked to a lot of the leaders in this area. Of course, we were at Blue Rock, who's doing their own version of cell therapy for Parkinson's, stem cell derived. There's other folks in Europe who are doing amazing work, Malin Parmar and Agneta Kirkby, for example, have their own approach for treating Parkinson's using stem cell derived dopaminergic neurons, for example. But this is autologous, and this is the ultimate dream for the iPS field is, of course, autologous cell therapy, not just for like cardiac myocardial infarction, for example, like what the field that I work on, but it's autologous cell therapy, autologous iPS therapy for Parkinson's. Okay, you generate a patient's own iPSC-derived dopaminergic neurons and do some sort of correction, and you can reintroduce them back into the person, ultimately repairing and ameliorating the disease. That's a dream, right? Very hard to accomplish that, not just because of science, but also because of the financial side of it, which we're not going to dive into too much here. But what they're doing here, and this is work that's, I believe, coming from MGH. Yeah. Harvard Medical School. I believe this is Department of Neurosurgery at MGH. Last author here is Kwang Soo Kim. First author here is Jeha Jeon. We're focusing on iPS-derived midbrain dopaminergic cells, which in their words represent a really promising source for potentially autologous cell therapy in Parkinson's disease. But standardized regulatory criteria, of course, are really critically important for clinical translation. Whenever you're talking about any pluripotent stem cell derived therapeutic product, you got to worry about tumorigenicity. You got to worry about efficacy, of course, safety of the delivery, all these kind of things that folks like Blue Rock are investigating. And this is a preclinical study, just to be clear, but they generated multiple clinical grade IPS lines, human IPS lines, from freshly biopsied fibroblasts of 4 sporadic Parkinson's disease patients using episomal reprogramming, and then differentiated them into these MDACs, these midbrain dopaminergic cells, using a refined 21-day protocol. So not too bad, right? And then they did a bunch of rigorous evaluation, as you might expect, with whole exome, whole genome sequencing, RNA sequencing, also some in vivo studies in mice that included a 39-week GLP, good laboratory practice, compliant mouse safety study. And so while these MDACs all met the safety criteria, the MDACs or midbrain dopaminergic cells from one patient failed to improve the rodent behavioral outcomes, which underscores the variability from person to person, potentially, or maybe in line to line, right? One thing they also alluded to was that their in vitro assessments didn't reliably predict their in vivo efficacy. In vitro, in vivo don't always have that one-to-one correlation, and actually identifying the dopaminergic fiber density as a key efficacy criteria that they have to look at. So ultimately, it's hopefully setting up a clinical trial. These findings are supporting quality control guidelines for autologous cell therapy, maybe paving the way for a clinical trial with their goal of 8 sporadic Parkinson's disease patients, which is scheduled to commence in 2025. And I think there are still a number of questions here. One being, the autologous part of cell therapy for these individuals. Do you want to be reintroducing an iPS-derived Parkinson's or iPS-derived dopaminergic neuron cell type dopaminergic cells that they describe here that have that ancestral genetic defect of the person itself that you're trying to restore, right? I don't know if you have to do some sort of editing to correct that mutation or correct something to going on, but I'm sure there's more to it that I'm really not diving super deep here. But like I said, this is one of the dreams of the IPS field ever since its inception in 2006, 2007, was generating a person's own iPS-derived cells for autologous therapy. And certainly a place like MGH, Mass General Hospital, would be able to support autologous, but I think the vast majority of approaches these days are going for the more allogeneic, off-the-shelf therapies like what Blue Rock is doing, in large part just because of the financials of it. Doing autologous cell therapy from IPS is just so expensive, even though that is, I think, the ultimate dream for personalized medicine in this field. What do you think?
No, I'm really torn on this, Arun. I just want to come out and say that this is a very important story. I'm so glad that they did this. Okay. I think it's pretty obvious and clear, and I don't want to belabor what you essentially just said, is that kind of moved on from this idea of autologous because it just doesn't really seem practical from an economic standpoint, mostly, right? Or a practical standpoint, having an IND for every single patient, right? Obviously, they could come up with a different like apparatus vis-a-vis FDA to get around that notion of an IND for every patient. But I think that captures the lift in terms of weight of the lift to do this for every patient. Now let's get back to why I think this is so great, because somebody has to do it. Somebody has to do the comp, right? We can't just move on. We kind of like never got off the ground. The whole autologous thing in Japan, one off with the patient, and then they were like, nah, backtracking. And there may be other conditions, I think, where the autologous thing makes more sense. And there may be a different like climate in terms of like the technology and the safety and the proofs and all that. But like right now, I just don't think it's going to get off the ground. But we need to see the comps. And what I think is so takes a lot of courage is to keep doing this while there can't be much like, industrial VC. I don't know that anyone's trying to pump a ton of money into autologous PD when you have Bayer and Blue Rock, right? But I think that the fact they came out and showed that, yeah, even one of these things it wasn't right, that didn't show the efficacy. These are all essential data points and moving in even to trials. I think maybe there's some people, and I think this is why I'm a little bit torn here. I think there's some people who will be like, what's the service to those patients to use this as opposed to, allogeneic? And I think that it's important because one, we all need to know, but also there could be a benefit to these patients. And while there is that idea of like introducing the genetically defective, yeah, sure, but like this is a terminal degenerative disease, right? So I think that having I just had an amazingly heartbreaking Someone very close to me told me that their husband was diagnosed with Parkinson's disease, and I would take anything. She would have taken anything. I'm calling people, hey, any of you guys doing these trials, listen, and please hit me up because it means the world to every one of these patients to have an option that is curative as opposed to palliative, I guess. It's a little harsh to say. Anyway, I'm really glad that this was done, and I'm glad that the trial is in patients. I don't think that this is going to be the future of cell-based therapy for PD, but this is a really important story.
Absolutely. It's about the hope, right? And that's what these major trials are doing, whether it's Blue Rock, whether it's the folks in Europe, it's delivering hope to patients afflicted with a disease that has such a terrible prognosis, right? And I think the autologous, while sure there's the financial considerations, you still can't take away that hope just because the financial implications may not be as great as the allogeneic, right? Like props to those folks at MGH. Like I think that's what we're both getting at here. Major props, major kudos to these folks at MGH who are pursuing this avenue in parallel with other folks around the world are doing. Really great stuff here.
Seriously, super props. And also, by the same token, I don't think a lot of other people could do it. You need a big, big spot like MGH with super high caliber investigators that can like risk the not so fruitful like backside of this in terms of the translational value. But yeah, great. Let's move on. We got another organoid story, as Arun alluded to in the jump. Symmetry today, guys. Symmetry, but all about hope. This is about the patient avatar idea, which I love. In this case, talking about individualized patient tumor organoids. Which, we've been talking about that forever. It's a great model. Avatar, you assess the drug response, this idea of personalized oncology. But on the ground, these models need improvement, right? The idea of it, to we put a tumor in a dish and it's just there, not really, like it grows. And you got to maintain that tumor microenvironment in order for it to, accurately mirror and predict the response of the native tumor in its native microenvironment, right? And that can be a challenge in terms of patient response prediction. We could do a lot better. And one of the major challenges is, that you don't accurately predict the drug responses of these tumors in patients. And that's the whole point, right? So the idea here, which is, I think, really clever and creative, it's from Shanghai, from multiple investigators. I'm going to highlight here and lead Younghae Wu, Haikun Lu, and Ying Mao at the State Key Laboratory, also at Shanghai Tech. And the idea here was to use tumor organoids, or to use normal patient cortical organoids as a vector, as like a host for tumor cells. And that's what the name of this story was, individualized patient tumor organoids, as they call them, these IPTOs. And the key here was in how they made them. They created them by not dissociating the primary tumor. So first they made the cortical organoids from the patient-specific cells, right? But then they preserved that tumor by not dissociating it, keeping the explant, keeping the native architecture, and then they insert them into these cortical organoids, or cerebral organoids. I don't know if they're cortical, but they use it as a host. Like, maybe it seems obvious now that I'm saying it, but I think that's so dope. And as you might expect, it accurately recapitulated that the cellular and molecular pathology of the brain tumors when they went in there. So they were able to check out multiple types of tumors: adult, pediatric, metastatic. They looked at all the omics. They looked at the histopathology, including epigenomics. I should highlight that. Like, they went across the genomic, right? They look at the genetic sequence, they look at the epigenome, also obviously transcriptomic, single cell, all of it. And they showed that the model really did a great job of mirroring the features of those original tumors. And here, critically, they were more accurate in predicting the patient-specific drug response and, wait for it, in a prospective patient cohort. All right, so here it is, game time. We're talking about personalized cancer therapy, at least in the case of these cerebral organoids, predicting brain tumors, which happen to be maybe the worst and the most intractable. So, like this, it's one of those things, Ruin, that I love, and I'm always surprised by with science, is that you talk about a thing and then you stop talking about it as much, and that's when the thing really hits. And this is it, I think, where the thing's really hitting. We haven't talked about the personalized medicine so much since we started talking about it, but now is the time that I think some of these therapies are trickling in and moving from theory to reality, and I think this is one that's going to be big.
As an in vitro modeler, this is mind-blowing stuff to me. This is that patient avatar realized, right? I mean, this is the dream. You have these cortical or whatever cerebellar brain organoids, and then you're introducing the tumor samples directly into the organoids, and then examining the evolution of the tumor in this model ex vivo, which you can potentially scale up into high throughput, and then bring in the drug testing side of it. I mean, the nuts to bolts quote clinical trial in a dish concept that Joe Brew is always talking about. This is the dream in the brain side of the field, right? I'm super excited about where this goes next. I think, there's always the disconnect between in vivo and in vitro. How close is the evolution of the tumor cells that you're happening in vitro to what's actually happening to the patients in vivo? Of course, like what we talked about a million times on the show here, it's a model, right? But this seems to be a very, very useful model. And one thing they actually alluded to here is the drug resistance side of it, because unfortunately, for a lot of these treatments in vivo clinically, patients ultimately develop drug resistance. Can these tumor organoids, hybridized organoids, whatever you want to call them, IPTOs, can they develop drug resistance in the same way as a patient would? Really amazing model here. And it seems like this is one of the only models that can actually preserve that intratumoral spatial heterogeneity of the tumor itself. So I love it. I can't wait to see where this goes in the years to come.
Oh, I love that you said that because you just planted the seed. Someone's probably listening to this and they're going to go have a Nature paper in about, I don't know, the way the review goes these years, about 7 years from now. But that's it, man. We can't emphasize that enough. You talk about these models, but you put it in a dish and it gets away from you, right? It's no longer the thing that you drew it from. So the fact that they can get they're making it even more complicated and yet reining it in and keeping it mirroring biology. And what you're just saying there, I think, is the big idea, is that to maintain it, like the next step is that you can see the evolution. And there's stories that have kind of done this, but I think here in a very like host, like using the organoid as a host, like legit host. I mean, minus the immune system and all that, but still, as a cellular host that evolves with the tumor, I think can get an idea to like tumor progression, the accumulation of multi-hit accumulation and precancerous cells. So this is kicking open the door with maybe what seems like a simple idea or straightforward or obvious in retrospect. But I think it speaks to the power of this field that we happen to be so lucky to be working in, Arun, is that these cellular organoids can do that. The fact that you can call a ball of cells a host for anything is pretty crazy, but I think it's apt in this case. So really big story, something we got to talk a bit about with our guest in just a couple minutes. Before we get there, though, I have a message from STEMCELL Technologies, and that is that you should learn how to perform embryoid body and monolayer-based neural induction of human pluripotent stem cells, or improve your existing neural induction workflows with a free on-demand course. To learn more and register, visit www.stemcell.com/NE-on-demand. Hey, everybody. On today's show, we have a special guest from the West Coast. I'm feeling very envious, jealous of Arun and George's situation over there as I'm snowed in Vermont, but I really can't complain. Howdy here is crazy. But as I said, we have a special guest on the show today, not just making me jealous, but talking about her lab's research focus. Dr. Giorgia Quadrato's assistant professor of stem cell and regenerative medicine at University of Southern California. The Quadrato Lab's focus is understanding the cellular and molecular basis of human brain development and disease. By combining the use of emerging models of the human brain with single-cell omics approaches, the lab is aiming to identify cell type specific disease mechanisms and, above all, new treatments for human neurodevelopmental disorders. Giorgia, thank you so much for joining us on the show today.
Thank you, Daylon. It's a real pleasure to be here, and thank you for hosting me, for giving me this opportunity. I feel like in the current climate is even more important to communicate science with the public. So thank you again for this.
Yeah, of course. I totally agree. And thanks for being on the show. I mean, you've been involved in this field of organoid biology for a while now. Of course, you've in particular focused on your pioneering work in brain organoids, and just down the road from me here in LA at USC, the University of Southern California. I mean, organoid biology has become so accessible these days, more accessible than ever to those of us in the stem cell field. And it's certainly caught the public's attention, like what you're alluding to. So to start things off, could you just give us an overview of what your lab is working on right now, especially as it pertains to organoid biology?
Yes. Yes, as Daylon mentioned, we are really focused on try to understand and study human brain development and disease. In particular, we are study two distinct brain structure. One is the cerebellum and one is the cerebral cortex. These structures are very interesting because if we think about evolution, these are the two brain structures that have evolved the most, especially if we compare great apes and humans. So this means that these structures are important for the acquisition of human-specific traits. And so it is, we are really interested in understanding neurodevelopmental disorders, and so how perturbation to development of these structures may lead to development of neurodevelopmental disorder. Specifically, we are interested in autism spectrum disorder and intellectual disability. So the mission and the research focus of my lab, actually, we have two main areas of focus. One is protocol development. So we are trying to increase the physiological relevance of brain organoids. And the other one is disease modeling. So they are obviously, these two parts are communicating. These are two distinct missions. And I have in the lab different people that are some lab members are more dedicated to protocol development and some others to disease modeling. What I love about being in the Los Angeles area is that we have the possibility to interact with other universities. So I feel like it's a very up and coming area. Obviously, we have UCLA, Cedars-Sinai. We are very close. So I'm in the medical school of USC, in the Keck School of Medicine, and we are very close to Caltech. And I'm very excited about this because we have many collaboration with the engineers, and especially in the field of tissue engineering and bioengineering, synthetic biology. So as a lab, we are really trying to integrate these technologies into brain organoids. And so we are experts in that. We are developmental biologists, but with the help of these other experts, we are trying to make, as I said, this model more physiologically relevant.
Yes, and it has never been more relevant, your model, and also, just stem cells generally, to the clinical applications and impact. And one of the most exciting things that I'm envious of, I mean, besides the weather, as I already mentioned, but specifically in your field, in terms of like cerebellar neural organoids, you talk about collaboration, and one of the most exciting things I think in your field is the synergy. Once these models were established and reproducibly, as I think a lot of people have emphasized, that these models are really reproducible, not just faithful models, but you can go high throughput and scale up and still get this fidelity. And apart from that, all the creativity, all the hacks we talk about on the show all the times in the Roundup, all these hacks that people have figured out, all these complementary models that kind of come out of the woodwork to create opportunities and leverage the model in ways that you never even thought of. I mean, we just talked in the roundup on this episode about these patient-specific cerebellar organoids. I don't know if you saw the cell stem cell paper from a couple weeks back, but the cerebellar, I think a few days back actually, just came out online. These cerebellar organoids serve as like a host for these patient-derived tumors, this personalized avatar of disease. So just one example of all the creative ways that these models have been applied. It must be so gratifying for you and your peers to see this collective output that has followed from your own work in many cases, particularly because these kind of studies, like I just talked about, the patient-specific organoids, a real critical watershed there. And with other stories, I think, is that they really are clinically impactful. I think that we're moving into this realm now where stem cells are being used to treat and diagnose patients. So how do you envision, I know that's like a cancer story and modeling, disease modeling, you're more, neurodevelopmental and neurodevelopmental disorders and physiology, but how do you envision, or what do you think are the most exciting impacts of, clinically speaking, of these cerebellar neural organoids moving forward?
Yeah, so with our protocol, we've been able to do two things that were, let's say, new compared to previous literature. One has been, we've been able to generate specific progenitor cell types of the rhombic lip, that is one of the neurogenic region of the cerebellum. And these specific progenitors are human specific. They have not been found even in great apes. And they have been associated with the development of medulloblastoma. So being able to generate these cell types in vitro really give us the opportunity to study a bit more the pathogenesis of medulloblastoma and also potentially screen for therapies. So I think this is very impactful. And then the other advance of our protocol is to be able to generate and to really mature Purkinje neurons in an all-human system. And these Purkinje neurons, so we've been able to basically culture these cerebellar organoids for a very long time, up to, we have characterized them in the paper for 6 months, but we know we can culture them for actually over 1 year. And we see that these organoids are able to generate network activity, inhibitory, excitatory neurons talk with each other in this model system, and particularly Purkinje neurons become functional. So they also show electrophysiological pattern that are the same that we see in vivo. And we can do this in an all-human system. So this is very relevant because we know, for example, that murine models of neurodegenerative disorder like cerebellar ataxia, they are not able to recapitulate this neurodegenerative disorder. And we know also that these Purkinje neurons are actually affected in many other disorders, including autism spectrum disorder. So we finally have a model in which we can really try to understand the biology of these diseases and again screen for therapeutics. So I think this is, what's the most important clinical type of relevance.
Yeah, and in addition to organoids, and this is certainly like the age of organoids, as some people have called it, it's also undoubtedly the age of single cell, right? Because these are two technologies that intertwine so well. It's the age of single cell and these transcriptomic atlases as well. It's something we've covered on the show here a lot. It seems like every show we're covering a new single cell atlas paper. We've looked at stories, looking at atlas approaches to untangle human brain development or single cell atlases of neurodegenerative disease. And actually recently you contributed to a very important organoid-centric atlas in Nature, which described an integrated transcriptomic cell profile or atlas of human neural organoids. And there are actually a lot of pioneers in the neural organoid field who contributed to this work, in addition to yourself, as part of this human cell atlas organoid brain network. And a number of these folks had actually been on this show, like Sergio Pasca, for example. Other folks include Jager Camp, Barbara Treitlein, many others. So could you talk more about this new atlas for brain organoids, how it came about, and maybe its utility for the greater community?
Yeah, sure. So I have to say that effort was led by, as you mentioned, Barbara Treitlein, Camp, we were able to contribute with our cerebellar organoid dataset. But I was very happy to be part of this work. I think it's very important because the problem is that when we generate organoids, this, I guess it's true for any type of organoids, but I'm going to talk about brain organoids today. So for when we generate these organoids, we usually benchmark them with, for example, if I'm generating a cerebellar organoid, we benchmark them with cerebellar tissue. The problem is that if we generate some off-target cells, let's say cells of another brain region, for example, the cortex, we are not easily going to be able to see that these other cell types are present in our data set. In some cases, we can see that if there are very special markers that identify this region. But in other cases, when they lose regional patterning, regional marker that identify these cell types of specific brain region, they will basically, when we do single cell sequencing, they will co-cluster with the data set of the reference data set. So for example, if you're doing basically try to benchmark cerebellar organs with cerebellar tissue in vivo, even if we have other progenitor from, let's say, midbrain, they will go and cluster with cerebellar progenitors. So it is important to have a reference data set that doesn't only contain one brain region, but contains all brain regions of the brain, because these will allow us to really understand if there are off target. In fact, what's very interesting about this work is that we have basically discovered that a lot of protocols that supposedly were generating only, for example, cortical cells or only thalamic cells, they're actually generating other off-target cell types. And now when we benchmark them against the entire fetal human brain, we see that. So it is really important, and we are doing now this in the lab, thanks to this tool developed by Barbara Trotler-Greykamp and Fabian Theis. We are really using now this full data set to benchmark our organoids. Otherwise, we are just working with organoids, and we are not sure which cell types are contained in these organoids. There is an example in the paper in which they show that they went back to an old publication about disease modeling, and they found that basically in this publication, the authors were comparing organoids, control organoids that were mostly making one brain region. I don't remember exactly, but probably 4 brain. And then the mutant organoids were not actually 4 brain organoids, were generating cell types of other brain regions. And so basically they were comparing organoids from different brain regions and not control with mutant organoids. So obviously this is a huge issue. If we want to do disease modeling, we need to make sure that organoids are reproducible and are generating the cell type of interest. And so this is very important. And so I hope most labs will try to include this type of benchmarking in their pipeline of analysis. And I think even for other organoid types, this should be done, because these organoids are amazing. They're really able to self-organize, and we are able with morphogens to direct their regionalization, but they're not perfect. And some cell lines can escape patterning more than others. So it is key. It's incredible. And I mean, it's, it's not anyone's fault if, some earlier paper protocols, we didn't notice that we were making off-target cell types, but now we know, and we should do, make our best effort to just, generate organoids that are only of the target region of interest.
Yeah, of course. I mean, that's science, right? You move forward, and initially you have, a breakthrough discovery. And then as the years pass on, you get finer and finer resolution. And look what a long way we've come with organoids from the first demonstrations. I mean, most people, I think, talk about Hans Clevers when we talk about organoids, I think appropriately, because, tissue specific adult stem cell organoids. I think the first observation of this notion of 3D and self-organization, as you referred to there. But I read this ISSCR Q&A that was done that you participated in, and I read it for background. And I was really touched by your citing Yoshiki Sasai as someone who inspired you. I mean, of course, a legend. And as this field progresses, I think we need to be reminded at every turn what a seminal contribution Sasai made with these optic cup organoids, right? Among many other amazing advances in his repertoire, by the way. But I can remember where I was when I first learned about the optic cup organoids, because it really was truly at that point, it was like, what do you mean? How? Because the whole idea, the faith in the ability of the cell to self-organize, the stem cells to self-organize from a pluripotent cell in particular, I don't think it was there for most investigators. So for me, I remember being really inspired, and of course, gutted to hear the final outcome with Sasai. But I just want to hear from you. Obviously the link in terms of organoids is there, obviously, but I just want to hear you, if you don't mind, expand a bit on how his career, how his work impacted your scientific trajectory.
Yeah. Thank you for this question. Absolutely. I feel like probably my career will be very different if Sasai was not there and didn't do all that he has done for the field. He is the real pioneer of this organoid field, and is basically the first demonstration of these amazing self-organizing properties of aggregate of human pluripotent stem cells came in 2008 from his lab. And so they were the first one to really demonstrate that aggregate of human pluripotent stem cells can lead to these pseudo-stratified organized structure that resemble the human cortical plate. And from there, so this was in 2008. And then after, I think, many years, so in 2013, we have the also really landmark paper from Madeline Lancaster, in which she was able to also leverage this principle of self-organization to generate these all brain organoids, in which she was able to characterize multiple brain structure. And then in the same year, Yoshiki Sasai's lab in 2013, they were the first one able to use morphogens to direct the differentiation of brain organoids into one specific brain region. And so, yeah, obviously, these are landmark studies, and it's really incredible how Sasai has been able to do that without the technologies we have nowadays, because now for us it's easy to find morphogens, and because we can do screening and we have single cell technologies, we can characterize the cells. It is, to me, is just an absolute genius, like being able to have that type of intuition and being able to discover and to really have this fundamental discovery and invention. I really, it just amazed me. I know he was studying a lot, and there were a lot of observation from in vivo development and developmental biology that he used to inform his studies, but still I think he had the incredible instinct, and it's still I don't understand how he managed that. And so he really discovered this, as I said, incredible self-organizing property of aggregate of pluripotent stem cells. And what he discovered is that basically stem cells are really great at generate cellular diversity, and also they're really great at generating local microarchitecture. So these cells in vitro can do that. What they are not good at doing is actually they are not. So there are some extrinsic cues that are very important for correct anatomical development. So of these organoids. So for example, we know by now that these in organoids, for example, if we want to make cortical organoids or cerebellar organoids, we will not make only one neural tube. In these aggregates, we will see multiple neural tubes. So the number of organ primordia, he discovered that the number of organ primordia is actually dependent by external cues. So organoids cannot by themselves understand how many of these cortex to make, for example. And then obviously, because of these, all the macroanatomy is basically completely missing and aberrant. So we do not have stereotypic anatomy in these organoids, but they are really great at making cellular diversity, so mirroring the cellular diversity we see in vivo, and also at the level of micro architecture, meaning we know, for example, these brain organoids are able to make a ventricle, and then on the top of this ventricle we see progenitors, and on the top of these progenitors we see neurons. So this type of cortical plate is there, and this is by definition organoids. They need to have a bit of organization in terms of site architecture, otherwise they're not defined as organoids. So these are all discoveries by Sasai, so it's just amazing and inspiring. And every time I want to, I need some inspiration, I go back and read these papers and reviews. Just incredible. And one of my dreams is actually to do a sabbatical in Japan, and I know now the Riken Center is different, it's changed a lot, but I really would love to be there for a bit. So
I mean, I think we all would, and I'm super excited about that ISSCR meeting that's happening in a couple of years, end of next year in Kyoto, the 20-year anniversary of IPSCs. I'll definitely be there, and I'm sure you'll be there too.
Absolutely, yes.
Yeah, and so I think one thing we're talking about here is the reproducibility of a lot of these protocols, and I think it's similar to IPS, right? Reprogramming was a seminal discovery, again, by Japanese scientist Yamanaka back in 2006, 2007. And in the beginning, I guess not everybody believed that it was real until you actually adapted the protocol and used it in your lab and saw that, yes, this is reproducible. This is something that anybody can do, generate iPSCs. And I think a big part of what has made brain organoids so accessible is reproducible protocol. Similarly, I mean, that's really the key for all new biotechnologies, right? You have to reproducibly demonstrate the production, the application of organoids. And you've recently published this protocol, Nature protocol, describing the generation, long-term culture of human cerebellar organoids from pluripotent stem cells, again, in that same vein of developing reproducible protocols for the field. So talk a little bit more about this protocol, that work that was recently published, and I mean, more generally, the power of standardized protocols for organoid biology.
Yeah, absolutely. I want to also say that there is there are still limitations in terms of reproducibility that derive from the fact that different iPSC lines or ESC lines, even worse, they have different propensity towards generating and differentiating. So we know obviously pluripotent stem cells have the ability to differentiate into the three germ layer: mesoderm, endoderm, and ectoderm, and we know that different cell lines have different propensity towards different germ layers. So this, in turn, gives rise to some intrinsic variability. So when we differentiate these cells, if, for example, we do not have a line that differentiates well into ectoderm, we will have problem generating brain organoids that are reproducible. And so it's just increasing the level of variability. There is effort from Sally Temple's lab and other labs in trying to basically, certainly Madeline Lancaster lab, trying to reset the state of these pluripotent stem cells so that they have all similar potential of generating, basically of differentiating. So, and I think when that is achieved, we will really improve and decrease the variability in this protocol. Because unfortunately, we can do so much to generate reproducible organisms, but if the starting stem cells have different differentiative potential, our job becomes very difficult. So in terms of our protocol, what we know is that with cell lines that are able to make ectoderm without problems, we do have a very, for these lines, the protocols works very well, very consistently. So the cerebellum basically contains two different germinal zones. One is called rhombic lip, and the other one is a ventricular zone. The ventricular zone gives origin to inhibitory neurons, and the rhombic lip gives origin to excitatory neuron lineages. And so successful cerebellar organoids, they have the ability to generate both regions, and therefore to generate the complete cellular diversity of the human cerebellum. we do see this in our organoids. We consistently see generation of both germinal zones, and all main cell types are there. Obviously, some subtypes may not be there, but just because we know activity and other external cues are important for the refinement of the identity of cell types, but this is true for every organ, I guess. And so we do not see really all the subtypes that we see in vivo, but the major classes are definitely there. So we have been really careful, and the only way to assess reproducibility of organoids is to do single cell sequencing, because it's the only way to really assess the cellular composition. And then we have done also benchmarking, as I said, against the entire human brain. And so we have been able to see that I think up to 80% of these cells that we generate in our organoids are cerebellar, and then we have a portion that are medulla cells, which are also come from same progenitors. So they are very reproducible. As I said, this doesn't mean that all cell lines will behave in the same way, but we have had a few that are very consistent. The problem is that when you get a cell line that doesn't behave well, especially if it's a patient cell lines, for now there is not much you can do. And my suggestion in that case is just give up, because that's not gonna work. And until someone like, Madeline Lacasse Temple fixes the issue with iPSCs, will be, this will stay a problem.
Yeah, we're kind of talking about that in the roundup for this episode about, the real challenges of autologous iPS-based therapy fundamentally in terms of clinical translation. I think it's the same problem, right? Is that you have this inherent variability where it's not really practical to vet all these lines and find the optimal line for every treatment that you do, right? And that's, irrelevant, I think, to the biology and that the fundamental insight that can be gleaned. So I'm not saying it's not worth pursuing, but it is a challenge. And I think one of the major facets of this age that we're living in, it's so exciting, is that the clinical translation is really picking up steam. We just talked to old collaborator of yours on the show, and I say this because of reproducibility, right? We just talked about reproducibility, and that's the key. You need to have a product that is reliable and that's going to get through the FDA. We just talked to an old collaborator of yours on the show, Bruna Paulson, who went from Arlata Lab, like you, to leadership at Gamido. And industry and academia, as we know, have also always been kind of hand in glove, right? But as I said, we're in the middle of this pretty substantial shift in the equilibrium. And I see this when you're trying to recruit postdocs, right? You get a lot of postdocs who are like, you know what? Yeah, I'll think about it. But I got a lot, I'll be honest, I got a lot of options. I'm not Giorgia Quadrato, so I don't got the kind of juice where the postdocs are knocking down my door. But nevertheless, I'm sure you see it too, that there is a bit of a shift in terms of the opportunities are there, maybe even more so over there in the fine weather of California, where you and Arun are doing the work in the cradle of biotech innovation there. You talk about also all the collaborators, Caltech and all the major universities, but also all the VC, all the biotech companies that it pre-existed stem cells. So yeah, it's a hotbed. You're very academic focused, but you got your patents. I've seen them. So how does this kind of shift, global shift, manifest in your lab, in your life, in your career? You got a little side hustle, starting a company. Share with us, will you?
Yeah. Yes. Obviously, as you mentioned, in California, there are lots of opportunities in the biotech sector. I have to say, at USC, we are also very encouraged to to start our own startup, and many faculty have that. Some faculties have more than one. So there is. I think there are lots of opportunities in that space. I recently, we have also, obviously in California, we have the California Institute for Regenerative Medicine. And so me, a colleague of mine, Niels Lindstrom, that is also assistant professor in the stem cell department where I have my lab, we applied for this grant from the California Institute for Regenerative Medicine, and Arun actually is another director. He also got this award, and so we're establishing a shared resource lab, is called, that is an institute, a center for that basically has the goal of democratizing the use of organoid technologies, and in our case of single cell omics technology, and facilitate access to Californian for these technologies. And so there are several shared resource labs that have been funded by the CIRM, California Institute for Regenerative Medicine, last year. I think there are 11. I don't know, Arun, if you remember exactly, but
I think that's right. Yeah, 11 now.
Yeah. So and so basically at the moment I have this is the side hustle I have at the moment, because I have my plate full with the lab and this other center that we are trying to grow. But that is more so I'm learning some aspects about commercialization, and then it's a very different type of job, let's say. So I'm doing that. And I have also ideas. So with some other former colleagues in Boston, we are thinking also about, we are trying to find VCs to sponsor a new project and a new startups that will leverage brain organoids and AI technologies to do drug screening for neuropsychiatric disorders. So so that's all ongoing. And to me, so I have actually some brilliant trainees in the lab, and I see that many of them are actually are a bit, I have to say, many of them actually would like to stay in academia, but they are worried. They are really worried about the current, obviously, climate for funding, the competition, and especially in terms of funding. They see that there is this tendency now to fund large collaborations, and smaller lab are struggling to get funding. And to them, this seems I feel like they seems very overwhelming. So recently, actually, the first author of the Cerebellar Organoid paper, Alex Satamian, he joined the ARC Institute. That is, I think, a fantastic initiative here in California that doesn't rely on public funding for the majority. And so he has accepted after the PhD a position as a scientist there. And his job is basically, he's working in a facility to, and basically is continuing to do what he did very well in my lab, which is developing new models and basically they have these fantastic facilities in which they and these are sort of core facilities that then help also core members with their own research program. So I see there are I see that many of my trainees are they are excited by this hybrid type of solution between academia and industry. So ideally, they want to still have their freedom to do research that is interesting, but then in an environment in which they do not have to struggle for funding because they see, PIs being really, affected by this. And so this is a problem. At the same time, we see that also the biotech, industry is getting a hit now. And if the funding situation gets worse for university, this is going to just feed back on the biotech and private sector. And so, yeah, it's scary. I think, if I was a trainee at this point, a postdoc, I would also be quite worried. So.
Yeah, it's definitely an uncertain time, that is for sure. I think because we're in California, we're somewhat shielded by that, because like what you're alluding to, we do have such a strong biotech industry. We do have other institutions like the ARC Institute that have these new funding models. So I think there are other possibilities here that there may not be elsewhere. And honestly, that's part of the reason that I'm out here too, is, there's all these other opportunities to not just dip your toes in the ocean, but also dip your toes into biotech, to industry, all these other possibilities of collaborating. Like you mentioned, you're just down the road from me here in LA at the University of Southern California. It's about like 10 miles between Cedar Sinai and USC, but that can take about an hour depending on traffic. And you alluded to CERM. We actually just had JT, Jonathan Thomas, the new head of CERM, here on the show. CERM is a big reason why, California has become such a powerhouse in stem cell biology. But in addition to philanthropic support, which has made a big deal for institutes like Cedars and also USC, and USC itself has become a tremendous powerhouse. Andy McMahon used to be the head of the stem cell program for a while, and now Chuck Murray, who's a renowned cardiac biologist, is in charge. And we actually just had him here at Cedars for a talk. He's awesome. So talk a little bit more about USC in general, how it's a great place for stem cell biology and education too. Stem cell education, you actually have a great master's program. So talk more about USC stem cell.
Yeah. So as you mentioned, we are in the medical school of USC, but we have this great master program that is linked to our department. And so this is a program that Andy McMahon wanted to establish, and he left this great legacy. We have many of these students are actually deciding to stay and pursue a PhD then at USC, and often in the developmental biology program. And it's very rewarding for us faculty to have the possibility also to teach in the space of developmental biology and stem cell research. So it's so in this sense it's great, and all faculty in the department are involved in teaching, even secondary faculty. And then what I love about our department is that it's really full of junior faculty. So when Andy McMahon started the institute over, I guess now it's 11 or 12 years ago, he started hiring junior faculty mostly. And so it is a very energetic, warm environment. I love my colleagues, so it is a fantastic place to do science. We are trying to create more opportunities for trainees as well. For example, we have now a new program in which postdoc have also mentoring committee, and they because sometimes postdoc are forgotten, like PhD students have mentoring committee, but postdoc don't, and so they have official committees, and then we are trying to create for them more opportunities in terms of building soft skill, career development opportunities. And we have also many of us are also very active I'm part of the Diversity, Equity, Inclusion Committee of the department, and many faculty are actually involved in that. And we are really trying to create an inclusive, supportive, collaborative environment. So I'm very happy. That, along with the fact that I said that we are really close to Caltech, and then we have the possibility to collaborate with all these other great institutions in Los Angeles. It's hard for me thinking about moving. It's difficult. I have to say I'm quite happy. And then now with Chuck taking over the leadership, we is also trying to really move the department more in a translational direction. And we are, Andy McMahon. I joined the department because of him, and he recruited me. But I think I'm very happy with Chuck's leadership at the moment. So he's been here for about 6 months, but is fully settled, and we are all excited to have him. So I think things will continue to be really, we are on an upward trajectory if NIH doesn't cut our funding. But
well, you said it. I mean, it sounds like a future is looking bright for USC, or as you said at the end there, but brighter than it's looking for some people outside of California. Not least of which, I mean, as Arun mentioned, we talked to JT on a previous episode, and he really drove home for, you guys knew this already, but I don't know if our listeners, and I certainly didn't know, that the CERM is like a NIH, essentially. It's like a state level NIH. And the focus on all these things that you mentioned, diversity, equity, inclusion, also the mentorship and the development, the collaboration, the sharing of reagents, it's so thoughtful. and has been a resounding success. And I feel like California, in turn, is looking like a really great place to land for a lot of young trainees who maybe look around, see the new administration laying siege against the NIH and the funding apparatus for the entire state. So scary times, but I'm optimistic about the future. And if nothing else, we've got you and Arun and your ilk out there staying toasty in the sun, dipping your toes in the ocean and sharing great insights with each other in the world. This has been a really great conversation. I really enjoyed it. Before we let you go, we just have a couple of peripheral questions. The first one being, if you weren't a scientist, what do you think you'd be doing with your life?
Yeah, so I always like this question because whenever someone asks me, I think, what I would do if I could do everything over. And I always think I will do this, have the same career. I love this job. I really enjoy the intellectual stimulation, the challenge. I really need that. However, as you all know, this is a very all-encompassing type of work. And so something that I miss is really trying to be connected more with the emotional, spiritual, or creative side of my being, and so I miss, really, for me, creative expression is something that I really love, and I don't have much time to do that. I also not a very talented artist anyway, but so sometimes I think what I would like to do perhaps is being an art curator, so being in touch with this artistic world that at the end I think art and science are really complementary. And if we think about whenever there's been like times like the Luminism, where scientific progress has really expanded, so we know that also artistic art has flourished. And so I feel like these are both important aspects of being human. And so I just wish I had more time for that, but maybe, who knows, when I retire I will have more time for that. Yeah.
Well, you're doing your art every day, Giorgia. I mean, Arun and I talk about this all the time, how intertwined art and science is, and how there is that creativity in all of us, and how that really is what science is. It's a search for answers and creativity. And sometimes maybe if that's inside, you could turn that to the spiritual end and search for a different kind of answer or answers about yourself. But that creativity, I think that's common to all of us, even the ones with little talent for art itself, like myself. I can take a great micrograph and just stare at it for hours. Finally, what's the best piece of advice you've ever received, whether it's professional or personal?
Yeah, so I have to say, I received very good advice, but we mentioned actually Chuck Murray, the new director of the department. And so we recently had a conversation about a couple of weeks ago, and we were chatting, and he really told me something that is very simple, but I think very important, that the importance of focusing. And I think, it is very difficult with our job to stay focused. We have so many tasks, and we have trainees that everyone, they have their own ideas, but it's very important. And I feel like it's very difficult, requires a lot of effort and discipline. It's like a muscle you need to sort of train and develop. But it is, I think it's very easy for all of us to lose focus. But if I think it's very important to try to understand what is the most important idea that we want to pursue, because when we start spending time on the third or fourth best idea, we are taking time away from pursuing what is most important for us, what is most meaningful. And so I think it's important to try, first of all, to understand what's most meaningful and rewarding, and then try to do our best to really focus and and this means saying no to great idea, something that we found exciting, but I think that's it's easy to say, oh, this is a bad idea, I'm not excited, so I'm not going to pursue that. I think we need to learn how to, at least me, I need to learn how to say because I get curious and interested about many things, but I found that very, very important. And because of our work, the nature of our work, we are teachers, we are mentors, we are entrepreneur. We are a lot of things, and I feel like the time for deep focus, for example, for our work focusing on, it's always, less and less, especially when you become more a senior and you have more administrative work, for example, university. And so I feel like it's something especially now that I'm becoming more senior, it's something that I want to put more effort on. And I cannot tell you exactly what strategies I'm going to use, but it's certainly I see. And it's something very simple that Chuck told me, but I thought a lot about it, and I think it's important.
Yeah, well, you kind of got to retrain yourself, as you just said, at every stage, right? When you're a trainee or, a younger scientist, you just got to kind of focus on what of all your shiny ideas, you got to focus on the few that are the best chance of success or biggest risk reward, whatever it is. And then you go to the next level and you got to kind of retrain your brain to not just focus on, within the funding, and then you got to give a fair shake to all the trainees, and then, as you said, the administrative. So yeah, I struggle as well, and I'll bet that even Chuck himself, while he's given all this strong advice, I'm sure he's being honest, he's certainly got plenty of shiny things in front of him too. So he's probably projecting a bit, but I would listen to Chuck if he had something to say to me. Give me a call, Chuck. I'll take your advice any day. Georgia, I'll also take an interview with you any day, and can't wait to have you back on the show. I think this has been a lot of fun, a great perspective, I thought, for trainees and PIs and everyone. So thanks for a really well-rounded interview, and thanks for sharing, and thanks for talking a little about the personal end with society. I always love to hear all sides of a scientist as bright as yourself. So many thanks from Arun, myself, and our listeners.
Thank you, Daylon. Thank you, Arun. It was a real pleasure, and thank you again for the opportunity.
All right, you guys, that brings us to the end of this show. What a fun exercise in symmetry. My mirror neurons are kicking. Don't forget to subscribe to our newsletter at www.stemcellpodcast.com to get the show notes, including an episode summary and links to all the interview and Roundup papers. You can also reach out to us on X at STEMCELL Podcast or by email at info@stemcellpodcast.com with feedback or to suggest guests.